Hello everyone. So today we're going to go ahead and review protein synthesis. So before we continue and we actually talk about the different process, I want to go ahead and talk a little bit about the major components of protein synthesis. Okay. So one of those is going to be DNA. So just like we had reviewed last time, DNA is going to be a nucleic acid that's going to be used to store genetic information. Okay, so when I say that it's going to store genetic information, that basically means it's going to contain our genes. And genes are going to be coding for specific traits, just like our hair color, how tall we are, allergies, all that kind of thing. Okay, so DNA is going to be made up of nucleotides that are the monomers. So they're going to be that small building block that we use to create something bigger. In this case, it would be a nucleic acid. Okay, so DNA is going to be made up of the four nucleotides called adenine and thymine. Those two bond together. We're going to have guanine and cytosine. Those two are going to pair together. And this is going to be what kind of distinguishes DNA and RNA from each other. Okay, so remember that in DNA, we're going to have thymine, that's going to pair with adenine. Okay, so the reason that DNA is so important when we talk about protein synthesis is because DNA is having encoding for those genes, right? So that important information that's going to go ahead and give the directions to make polypeptides or proteins. Okay, so that's why DNA is so important in this process. The next thing that we're going to go ahead and review before we continue is going to be a little baby review on RNA. Okay, so like I mentioned, just like we talked about with DNA, RNA is going to be a nucleic acid, but this one's going to be really critical for protein synthesis because without it, we can't go straight from DNA to protein. So RNA is going to be like that middleman. It's going to be in the middle to kind of make sure that the message is translated between DNA and protein. Okay. So the major difference between DNA and RNA is that RNA is going to be single-stranded. So we don't have any hydrogen bonds between two strands. Here we just have one single strand of nucleotides. And instead of having thymine, we're going to have uracil. Okay, so adenine and uracil will pair together, and then guanine and cytosine will pair together. So we're going to have three different types of RNA that all have a really important role when we talk about protein synthesis. Okay, so the very first RNA that we're going to have is referred to as messenger RNA or mRNA. And the reason that this is really important is because this is what's going to take the directions from DNA and exit it into the cytoplasm. Okay, so when we talked about DNA, we talked about how DNA is only going to be present in the nucleus. So messenger RNA is going to take that information from DNA and take it out of the nucleus to find a ribosome. Okay, so mRNA is going to look like this here in this picture on the bottom right corner, where you just have like one single strand with nucleotides in it. So it's just going to be complementary to the DNA strand. So it's going to be the exact same message, just in RNA format. Okay, the second type of RNA that we're going to have is ribosomal RNA or rRNA. And ribosomal RNA is going to be really important because it's kind of what makes up ribosomes. Okay, so ribosome is going to be an organelle that's going to be found in all cells, and it's going to be what makes proteins. Okay, so ribosomal RNA is making a ribosome. Okay, and then lastly, the third type of RNA that we have is going to be transfer RNA or tRNA, and this is going to be like the machine that kind of brings the amino acids. Okay. So in the next slide, we're going to talk a little bit about proteins, but transfer RNA is essentially taking the monomer of a protein, which is an amino acid, and it's recruiting it, bringing it back to the ribosomal RNA. So it's kind of like a little like crane arm, kind of bringing back those amino acids to the ribosome. Okay. And then let's go ahead and review protein. Okay, so protein is going to be a biomolecule and it has a lot of diverse functions. Okay, so some of its functions are going to be to speed up chemical reactions. Others are going to be able to form our muscle cells, right? That's why when you do intense exercise or people who work out take protein shakes, right? Because protein is really important for building muscles. We're also going to be using proteins to transport molecules into the cell or even outside of the cell. And this is through that process of like active transport, right? As well as lastly, we use it to fight disease. So these type of proteins are referred to as antibodies, and I like to think of them as like our body's medicine. So it's like the medicine that your body creates to fight infections. Okay, so like I mentioned in the previous slide, the monomer for a protein is going to be an amino acid. And there's about 20 different types of amino acids, and that's why we can produce such diverse proteins, is because of all the combinations that we can make. Okay, so the monomer being amino acids, that's going to be the small building block that we're going to put together to create a polypeptide chain. Okay, you're going to hear that word often because a polypeptide chain is what we refer to as like amino acids that are bonded together, but that's basically just a protein, okay? So a polypeptide chain is just a fancy word for protein. And so if you can see here in this picture, right, this like little bead, it looks like a necklace, like a beaded necklace. That's going to be that polypeptide chain or that protein. And you can see that each separate color is going to be signifying a different type of amino acid that's bonded together using a peptide bond. Okay, a peptide bond is different from a hydrogen bond, so make sure you don't get those confused. Hydrogen bonds are important for DNA. Peptide bonds are going to be important for protein. Okay, so one of the first things that we're going to go ahead and talk about when we talk about protein synthesis is the idea of what's called central dogma. Okay, and central dogma is going to be the process of taking DNA to make proteins. Okay, so DNA, like I mentioned, cannot directly make proteins, and so that's why mRNA and RNA in general is really, really important for protein synthesis. Okay, so central dogma is going to be talking about the process of going from DNA to mRNA through the process of transcription, then from mRNA to protein using the process of translation. Okay, so transcription and translation together are going to be what's referred to as protein synthesis. And these two processes is what's going to lead from taking that essential gene information from DNA and taking it into a protein that can have any kind of diverse function from antibody to enzymes and that kind of thing. Okay, so again, DNA cannot go directly into protein, and so that's where mRNA is going to come in. Okay, so just like I mentioned, right, these two processes of transcription and translation are going to go ahead and work together to create proteins. And so the first step is always going to be transcription. And transcription is going to be taking the DNA, okay, and it's going to go ahead and make a complementary mRNA strand. Okay, when I say complementary, what I mean is that we're going to have like the nucleotide that pairs with whatever's in the DNA strand, it's going to be complementary, so it's going to match. So for example, if we have a thymine, then the mRNA would go ahead and bond with adenine. If we had an adenine, the RNA would go ahead and create a uracil. If we had a cytosine, the RNA would go ahead and have a guanine, and vice versa. Okay, so we're going to go ahead and taking that DNA, we're going to make a complementary, so matching pair of mRNA. After that process is complete, we're going to go into translation. And translation is the process of taking that complementary mRNA strand and then going ahead and making the polypeptide, which is going to be that protein. Okay, so we're going to go into a little bit in depth into each of these processes, but we're going to go ahead and start with transcription. So transcription is going to be starting off the process of making proteins. Okay, so when we talk about proteins and the function between them, I like to mention that they're gene expression. So if we think about a protein, it's really kind of abstract of an idea of like how they function, right? And why DNA is so important. But DNA is going to have genes, right? And those genes are going to be what's making proteins. So proteins are what's referred to as gene expression. So what's causing that gene to be seen. Okay. So transcription is the process of making mRNA from DNA. Okay, and this process is going to be happening in the nucleus. Okay, so like when I mentioned at the beginning with the DNA review, I mentioned that the DNA is never going to leave the nucleus. That's like its little home. Okay, so mRNA and the process of transcription is going to happen in the nucleus. Okay, and transcription has three separate processes and steps. The first is going to be initiation, so like the beginning of the process. We're going to have elongation, which is just the process of making the mRNA. And then lastly, we're going to have termination, which is where we're going to have the stop like of the production of the mRNA. Okay, so an important enzyme that's going to be allowing for all this to happen is going to be the RNA polymerase. So RNA polymerase has two jobs in transcription. The first is that it's going to go ahead and unwind and unzip, so basically open up the DNA strands. So it's going to be breaking the hydrogen bonds between the two strands of DNA, and then it's going to go ahead and separate them so that it can go ahead and start making that mRNA. Okay. The second job that the RNA polymerase is going to have is making that mRNA. So it's going to be making the complementary strand to the DNA in order to have the end product of mRNA. Okay. So when you think about transcription, what you really need to remember is that the product is going to be mRNA. Okay. So this is kind of like what I was mentioning here, right? So the RNA polymerase is going to have those two jobs. So like I mentioned, the first is opening up the DNA. So these red and blue lines here are representing the two separate strands of DNA. So it opened it up. You can see that now we have this little like opening here for the RNA polymerase can go on ahead and work. And then the second job is that it's taking the complementary nucleotides and creating the mRNA strand that's in purple here. Okay, so we have the template DNA strand, and we're going to have free RNA nucleotides that are going to be put into bonded together to create that mRNA. Okay. So after we have the production of mRNA, we go into the process of translation. Okay, so translation is going to be that second step in making proteins, and it's going to be making proteins from that mRNA. Okay, and this process is now going to happen in the cytoplasm. So once mRNA is created, it leaves the nucleus. Okay, and one of the perks about mRNA is that it's much smaller than DNA, so it can leave the nucleus and it's going to go into the cytoplasm. So this is really important because in the cytoplasm is where we have the presence of ribosomes, which are going to be that organelle that's going to start off and make proteins. Okay, so translation is happening in the cytoplasm, and it's going to require several things. Okay, so we're going to require ribosomal RNA. Right, so remember that when we talked about RNA, we talked about how ribosomal RNA is going to be making up ribosomes. It's going to require transfer RNA. So that's going to be what brings in those amino acids to build the proteins. Okay. And then lastly, we need that building block. We need the monomer of proteins, which are the amino acids. Okay. So similarly to transcription, translation is going to have three separate steps. So we're going to have initiation, elongation, as well as termination. Okay. But as a whole, okay, the end product of a protein of translation is going to be proteins. Okay, so transcription was mRNA, translation is proteins. Okay, so here in this picture, I have all the different steps, right? So I mentioned the three steps of initiation, elongation, and termination. So the process is going to happen when mRNA finds the ribosome, right? So remember the ribosome is going to have the ribosomal RNA. So those two are going to join together and they're going to start off that process, okay? What's going to happen is that during the elongation stage, okay, tRNAs are going to bring in amino acids to start building that protein, and the ribosome will build peptide bonds between those amino acids. So you can see, right, we had this little pink one kind of enter, and you can see that it has, well, the process actually started off with the blue one, right? So the blue one entered, okay, and it moves along into the steps, and then we had the pink one enter, and now we have two separate amino acids that are bonded together, and then the process is going to continue until we have reached the codon that's going to stop the whole process. and we're going to have a polypeptide chain. Okay, so that polypeptide chain is what's created here, and termination is the process of ending that and cutting the polypeptide. Okay, so the last thing that we're going to go ahead and talk about is what I just mentioned, which is a codon. Okay, a codon is going to be a set of 3 nucleotides, and they basically determine the amino acid sequence. So they kind of determine which amino acid is going to be brought in by the transfer RNA to create that polypeptide chain. Okay, so here at the bottom of the screen, I have an mRNA, and I basically have all the nucleotides labeled below, and I have them already in sets of 3. Okay, so each set of 3, as you can see, is in a code for a specific amino acid. Okay, so that's what the transfer RNA is going to go ahead and recruit. And this is really important when we talk about amino acid sequences and proteins. Okay, so before I go ahead and kind of end today's video, I want to talk a little bit about a codon chart. Okay, so this is a type of question that you'll definitely see on the STAR exam. And really, they're pretty easy. You just need to learn how to read the table. Okay, so here you'll see this table on the STAR exam, and it has 3 separate components. So you have the first letter, the second letter, and then the third letter. First, second, and third letters are referring to each letter in the codon, right? So remember, a codon is going to be a set of 3. So let's go ahead and try with 3 different examples, right? So here we have the first letter. We have this codon that's AUG. First letter is A. All right, so I'm going to go down to this section here. First letter, A. Second letter will be U, so AU, so it would be in this box. Third letter G, right? So AUG here. This is going to be our start codon for methionine. Okay. Then let's go ahead and try with the second example. So we have the first letter, which is going to be U, so up here. Second letter is going to be U as well. And third letter U. So we're going to have the codon over here, which is going to be for this specific amino acid. Okay. And then lastly, let's go ahead and do the third example. So we have GCA, right? So G being the first letter, C going to be the second letter, so in this column here, and then A being the last letter, so it's going to be for alanine. Okay, so that's how you would read a codon chart. Make sure that you always know the first, second, and third letter, and kind of find the specific codon that you want. So this can be a little bit tricky, as you can see that for like the third example, right? Everything that has that's in that square is going to code for alanine. So just make sure you're paying attention to the details when you're trying to answer these types of questions.